topic of this video is proteins, which are organic compounds that are composed mainly of carbon, hydrogen, oxygen, and nitrogen. Nitrogen is what sets proteins apart from the other classes of molecules that I'm going to talk about in these videos. Proteins are the construction materials for body parts such as muscles, skin, and blood.
Now we approach the most complicated, powerful, polymorphously awesome chemicals in our body, the protein. And by complicated, I mean that they are probably the most complicated chemical compounds on the planet. In fact, they're so amazing that we're going to do a separate episode on them and how they are created by DNA. But right now, in you, there are tens of thousands of proteins doing everything they can to keep you alive. There are enzymes regulating chemical processes, helping you digest food. There are antibodies connecting themselves to invaders like bacteria and viruses so that your immune system can get them. There are protein endorphins that mess around with your brain and make you feel emotions. They're everywhere. They do everything.
Okay, they're everywhere. They do everything. So the monomer for proteins is a molecule called amino acids. Amino acids, we have 20 of them in our body, but those 20 amino acids make up the tens of thousands of proteins and enzymes and antibodies like he talked about in the video. And I will explain a little bit later how you can use just 20 amino acids to make tens of thousands of different proteins.
Just like fatty acids, amino acids have a carboxyl group at one end, and on the other end, they have an amino group, amino acid. Now, hey, I don't know if you noticed this, but this is the first time that nitrogen has shown up in our food. This is super important because despite the fact that nitrogen is everywhere, it's 80% of the air, we can't just pull it out of the air and put it into our bodies. We have to get nitrogen from food, and so we have to eat foods that are high in protein, like this egg, which by its very virtue, because it is all the white part is protein, it contains a goodly amount of nitrogen. Now, in the middle of the amino and the acid group is a carbon, and it shares one of its electrons with good old hydrogen, and the other electron is free to be shared with R, which is just to kind of fill in the blank. We call it the R group.
Okay, so this is the structure for all 20 amino acids. And I'm just going to go again to kind of repeat what he was explaining. So there's a central carbon. This is the only thing, by the way, which is different for all 20 amino acids. Otherwise, all 20 amino acids have exactly the same structure, which you see here in the top half. So you have a central carbon, and it doesn't matter which side or which order it goes in, but on one side of the carbon, because remember, carbon has 4 electrons in its outer shell, so it wants 4 more, so it forms 4 single bonds. One of those bonds is going to be to a lone hydrogen atom. One of those is going to be shared with an NH2 or amino group. Remember in the last video I talked about functional groups. Here you see a lot of them. And one of those bonds is going to be to carboxyl group, which is COOH, a carbon double bonded to oxygen, and a hydroxyl group. Now, for the fourth side or the fourth bond of this central carbon, we have what's called the R group. Now, the R group is different for each of the 20 amino acids, and this is what is going to contribute to us being able to use only 20 amino acids to build tens of thousands of proteins. So, as I said, amino acids are different only in the type of R group they carry. The difference among the amino acid R groups give different proteins very different shapes.
Must be called a side chain, and there are 20 different kinds of side chains. Whatever fits in that blank will determine the shape and the function of that amino acid. So if you put this in there, you get valine, which is an amino acid that does a lot of stuff like protecting and building muscle tissue. And if you put this in there, you get tryptophan, which may be best known for its role in helping you regulate mood and energy levels. Amino acids form.
Okay, so he just went over a couple of examples here. I've got all 20 amino acids, and highlighted in the different colors are the R groups. Now, what this diagram does is it actually divides them up into their properties. So you can see that for 10 or half, oh, no, sorry, it's nine. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. For 9 of the 10 amino acids, they have non-polar side chains. So you can look at each of these each of these side chains, they're non-polar. You don't see any OH bonds at all. Remember, OH bonds are what contribute to polarity. Here you've got polar side chains, so you're going to see OH. So you OH here, there's OH. Sulfur is right below oxygen or above oxygen, I can't remember, but it's in the same group as oxygen. And so remember from chemistry, actually, the homework that you did last week, it talked about elements that are in the same group have the same chemical properties. So sulfur is going to have similar properties to oxygen. So that means sulfur hydrogen bonds are also going to be polar. And here's another OH. I think the NH2, the amino group here, gives it polarity. Anyway, so these are the polar side chains. And then you have these down here, which actually carry with them an electric charge. So you imagine if you take these 20 amino acids and if you put them in different arrangements, because of the polar side chains and electrically charged side chains, you're going to get some forms of attraction and some hydrogen bonding going on there. So depending on the order of the amino acids, the chain is going to form hydrogen bonds in specific ways, and that's going to create a specific shape to the protein. And I'm going to say this probably a million times while I'm your teacher, is that shape determines function. I use the phrase form follows function. The shape of things is because of the function of those things. And so proteins completely rely on shape for their function. Okay, so again, with a condensation reaction, we're going to remove hydrogen from one and hydroxyl from another one. We form a bond between the two amino acids and we create a water molecule. For proteins and only proteins, the bond that is formed has a specific name. It's called a peptide bond, and the prefix pep indicates protein. So again, with vocab, pep, protein. And then we put polypeptides together using long chains of amino acids, and whole proteins can be made of one or more polypeptides. These chains are so long that they fold and twist from those hydrogen bonds that I talked about because of the R groups. And so the sequence of amino acids is going to determine how that folding and twisting occurs, and therefore it's going to determine the shape of the protein.
Amino acids form long chains called polypeptides. Proteins are formed when these polypeptides not only connect, but elaborate in, frankly, really elegant structures. They fold, they coil, they twist. If they were sculptures, I would go to the museum every day just to look at them, and I'd walk straight past the nudes without even looking. But proteins...
Okay, so I'm going to cover how the protein actually comes together in its shape. There's four layers to a protein, or 4 levels, 4 layers, whatever, to protein structure. So we have primary structure, secondary, tertiary, and quaternary. So primary structure results from the sequence of amino acids that can make up the protein chain. Like I said, it's just literally the order of the amino acids, like what amino acids go in which sequence. Secondary structure is where you start having those hydrogen bonds, which is going to make the string of amino acids kind of fold or twist. Oftentimes, this results in a helical or a spiral shape. That's secondary structure because of the hydrogen bonds between the R groups. Then we get to tertiary structure, or third level. This is the 3D shape or conformation of a protein. This structure, the tertiary structure, most determines the way that a protein functions, a 3-dimensional structure or shape. And finally, quaternary structure only refers to proteins that are made of more than one polypeptide chain for an example, hemoglobin, which is the protein found in red blood cells, it is made of 4 polypeptide chains, which you can see here in different colors. Each hemoglobin, you can also see each hemoglobin has an iron atom in it, here and here. And so every red blood cell will have probably tens of thousands or hundreds of thousands of hemoglobin molecules to carry all that oxygen, because iron is the atom that oxygen physically binds to be carried in the red blood cell. So this is going to be an animation showing you these 4 levels of protein structure.
Proteins are building blocks that help give your body structure and do work around your body. They move molecules, they make new molecules, they recycle old molecules, and this is just to name a few of the things that they do. But maybe the most interesting thing is that these protein building blocks are themselves made up of smaller building blocks called amino acids. Here you can see a string of amino acids. The different shapes that you can see represent the atoms that make up each amino acid.
This is primary structure, by the way.
And here we've highlighted 12 individual amino acids. To make it simpler, each different amino acid can be represented by a single letter.
So each letter is representing a separate amino acid. So like I said, this represents primary structure. It's just the sequence of amino acids.
Now each amino acid is shown as a colored ball, looking like beads on a string. This makes the protein structure easier to imagine. The order of amino acids is only part of the story. Because of the different shapes of the individual amino acids, they like to fold into even more interesting 3-dimensional shapes.
This is secondary structure.
This molecule is twisting into several different spiral or helical shapes, and then those are folding on each other. Take a look at the 3-dimensional shape as we give the protein a spin. Kind of looks like logs stacked in a fireplace. Here is the 1-letter amino acid code revealing the identity of each amino acid. Again, now you see the amino acids drawn to show the position of each atom. This is like looking at an atomic skeleton of each amino acid.
And finally,
just like you
tertiary
take up
structure.
more space than just your skeleton would, see how much space each atom really occupies.
So this represents tertiary structure, which is the 3-dimensional shape of the protein.
This is the real shape of the protein. Now we've seen an example of a protein taking shape, and several of the ways scientists visualize these tiny building blocks used throughout your body.
Okay. Now, a specific class of proteins are called enzymes. Most enzymes are proteins, so it's important to remember that enzymes are proteins, but proteins aren't necessarily enzymes. They're just kind of like a subclass of protein. The function of the enzyme is to break down large molecules into smaller ones. It used to be called the lock and key model, but now it's called the induced fit model. And this is why shape is so important for proteins, because an enzyme only functions if it has a physical fit with the substrate. The substrate is the substance that's going to be broken down. And you can also see that, so you have the substrate here, this is your enzyme. You see that they have to physically fit together. So this enzyme wouldn't actually work, because if you look at this part, it's square at the bottom, but wait, my iPad just quit. Okay, so the substrate, you can see that the shape would not fit with this particular enzyme, because this square part would not fit. Why is that white? Oh, wouldn't fit with this round part here. And you also see that this part is much deeper than that part of the enzyme goes. So this actually wouldn't work. This wouldn't work. And that's called the induced fit model. It works just like a lock and a key, which is why it used to be called the lock and key model. And the last thing, remove all of that crap here. The last thing is where the substrate and enzyme fit together is called the active site, which you can see here as well, the active site. And then the enzyme will break down the substrate and then it can be reused over and over again. So again, the active site where the substrate binds, because each enzyme is specific for the substrate, only one type of enzyme can break down protein. There's another enzyme that can break down carbohydrate, etc. And it even gets more specific for, let's say, carbohydrates. You have some enzymes, like I mentioned in the carbohydrate video, the salivary amylase that's in your saliva. It is made to break down starch into maltose, and you'll have separate enzymes in your small intestine where the maltose is then further broken down into glucose. So everything in your body that gets broken down has its own unique enzyme. Okay, that's way too long. Right, so the last thing about proteins is that there are two things that affect protein activity, and of course, including enzyme activity, that is temperature and pH. But I want to draw your attention to the shapes of these graphs. They're very different. You can see for pH, the slope up and the slope down are equal. So most enzymes work at an optimal pH or an ideal pH of around like 7.5 or 8. Pretty much a neutral pH, except the enzyme that is produced by your stomach, because your stomach has hydrochloric acid in it. That's a pH of 2. So the enzyme in your stomach will not work at a higher pH. It's specifically made. It's actually it's a protein that is secreted into your stomach, and as soon as that protein hits the acidic environment, it's converted into an enzyme that breaks down protein. But you'll learn all about that next year. The big difference in the temperature graph that you see that it slopes up very gently, but then at a certain temperature, it drops off very abruptly and very quickly. And there is a reason for that, which I will talk about. If the temperature becomes too high, and it's normally around, I think, 41 or 42 degrees, the enzyme starts to break down. And how it breaks down, it's a process called denature or denaturing. Denature basically just means to kind of fall apart or unravel. Going back to, okay, get out of there. Going back to, my iPad is slow. Going back to here. And okay, so you remember how the amino acids, they form hydrogen bonds in their secondary structure. No, there we go. And their secondary structure, and they fold like this. At high temperatures, those hydrogen bonds that are holding the protein in shape are going to start to break.
To several different spiral.
Okay, she can be quiet. So all of these hydrogen bonds that are holding these chains of amino acids in its particular shape, those hydrogen bonds are going to start to break down, which means your overall shape of your protein is going to break down. It's kind of like, I don't know if any of you guys knit. I knit things. But it's like if you have a knitted sweater and you have one string that comes loose, and then basically you can just unravel the whole sweater. That is how denaturing works. So that's why the graph, the temperature graph, it drops off suddenly. And a really easy way, like you guys have seen protein denature probably like every day of your life, like every time you cook an egg. If I asked you to describe what an egg looks like when it's raw versus when it's cooked, and you'll probably say, well, the egg white when it's raw is actually clear liquid. And then during the process of cooking, it turns into a white solid. And that's because the egg white is made of 100% protein called albumin, just in case you feel like being smarter than people around you. Albumin is the name of the protein. By the way, the suffix "in" when you're talking about molecules in the body, that says protein. Ooh, my pad just fell. It scared the crap out of me. the suffix in means it's a protein. And so when you cook the egg white, you denature the albumin, and that's why it changes into a white solid from a clear liquid when it's raw. And scientists, actually, I read an article, I'll have to see if I can find it again. Scientists are actually working now to figure out how to, I guess I'd call it renature, how to reform those hydrogen bonds so that you could basically uncook an egg. You could turn it back into a raw egg, because all it is denaturing, which would be super interesting. Okay, so that's why when you get to a certain temperature, the enzyme activity drops off sharply because of denaturing, and the protein just falls apart, the enzyme just falls apart. And for pH, again, it's more of a gradual, it works better and then it starts to not work as well at pHs that are too high or too low.
Protein molecules. They speed up reactions.
Sorry, let's go back.
Enzymes are usually large protein molecules. They speed up reactions. Imagine this is amylase. It helps break down starch into smaller maltose molecules. Without an enzyme holding the large molecule in position, this reaction would be extremely slow. But is the activity of salivary amylase affected by temperature? A measured amount of starch solution is placed into 3 test tubes. These are immersed in water and are kept at different temperatures, 5, 20, and 35 degrees Celsius. The same amount of salivary amylase is then placed in each. As soon as the amylase comes into contact with starch, it'll begin to break the starch down. A spotting tile containing a reddish-brown solution of iodine will help monitor the reaction. After 1 minute, a drop of each solution is added to the iodine. First the one at 5, then at 20, and finally the solution at 35 degrees C. Iodine turns blue-black if starch is present. Another minute later, and the solutions are tested again. This time, the amylase in the warm solution has already converted all the starch into sugars. As time goes by, the starch in the other test tubes is gradually broken down. After 3 minutes, starch only remains in the coldest solution. It may be several minutes before a change is observed at 5 degrees C. At what temperature does the action of amylase appear to work best? Enzymes are made of protein. If the surrounding temperature is too high, the molecules denature. The shape of the protein changes. See what effect this has on its activity. What might happen if the amylase and starch solution is kept at 60 degrees Celsius?
If it was kept at 60 degrees Celsius, the enzyme would completely break down and you wouldn't have any starch at all in the solution. And that is proteins.